Thank you, Swati, for a warm welcome. Can you just enable me to share the slide?
Yes, Dr. Austin, I think you are the host now, so you can take over.
Okay, thank you. Am I visible? Is my slide visible to all? Okay. Thank you. Uh, I'm audible? Uh, Mm. Mm. Is it visible now? Can you see the slides now? Right. So as already Swati has introduced me, let me just start by introducing myself. I'm Dr. Afreen Inam. I'm an assistant professor at the Department of Chemistry, Jamia Millia Islamia. So today I will be talking about medicinal chemistry and rational drug design for precision and personalized cancer therapy. So, first, any field which is related to drug design or medicine comes under the domain of medicinal chemistry. So, let us start with a brief introduction of medicinal chemistry first. Medicinal chemistry is not a standalone field, it is an interdisciplinary science which focuses on discovery, synthesis, and development of new drugs. And it brings together different domains like organic synthesis, biochemistry, chemical biology to create or modify molecules, to biochemistry, that is for disease mechanism and molecular targets, pharmacology, pharmacology to new drug interactions, toxicology to ensure safety, and computational studies to predict and optimize drug behavior even before the synthesis. This integration of diverse disciplines, this is what enables the medicinal chemistry to move from a biological idea to clinical useful therapeutic agents, making it central to rational drug design and precision medicines. With this overview, let us explore how all these components work together in drug discovery process. So first of all, let us talk about what is the concept of drug designing. Drug designing is a systematic yet creative process in which new drugs are designed on the detailed knowledge of the structure of the target, such as enzymes, receptors, and proteins. It is the core component of drug discovery and emphasizes on rational planning and optimization rather than trial and error methods. A key focus of drug design is the understanding of molecular interactions between the drug and its target, while we simultaneously optimize physiochemical properties such as solubility, stability, and bioavailability. Thus, the drug design represents a balance between scientific reasoning and innovation, which is aimed at developing effective therapeutic agents. Now, this slide highlights the scientific scope of medicinal chemistry in drug discovery. Okay, so here, despite significant advances in the rate of new drug discovery has slowed down, which is due to the stringent safety and efficacy requirements, complexity of rational designing, and the high cost of research and development. But still, medicinal chemistry supports drug design by, although it is being very challenging, still medicinal chemistry provides a chemical foundation for drug design by focusing on identification and synthesizing a systematic optimization of biologically active molecules. These molecules may originate from natural drugs, screening libraries, or computationally designed scaffolds. Beyond synthesis, medicinal chemistry is deeply concerned with molecular mechanism of drug action, which includes how small molecules recognize and bind to the biological targets, the nature of their key interactions at the binding site, and how structural modifications translate these changes into biological activity that we describe as structure activity relationship. The ultimate objective of this discipline of a medicinal chemistry is what? It is the discovery of novel therapeutic agents, achieving which requires interdisciplinary skills. So a medicinal chemist must integrate advanced organic synthesis along with biochemical understanding of the disease pathways and pharmacological evaluation of the drug responses. In this way, the medicinal chemistry act as a bridge between the converting the molecular structures into clinically relevant medicines. So before we get into the depth of medicinal chemistry and drug design, that is, let us just get aware with some of the terminologies which are used commonly in the medicinal chemistry. Most of you, all of you must be very much well aware of all these terms. I'm just giving a brief uh, view. So first, what we are talking about drug. What does that mean? It is a chemical substance that has already been proven to be safe and effective in humans for treating diseases. However, in early research, we don't call it as a drug, we call it as a hit. A hit is simply a molecule that shows some measurable activity in initial screening tests. Through these chemical modifications and optimization, the hit can be improved to a lead, which is further selected for optimization. If it shows better affinity, acceptable biological properties, it becomes a drug candidate suitable for clinical trials. Most drugs act by binding to specific biological targets, which are known as receptors, and depending upon how they interact with these receptors, they can be act as agonists or antagonists. That is either producing a biological response or they are blocking that response. Once we have a potential drug candidate, the next step is to understand how well and how safe they are. So potency, it tells us how much drug is needed to produce that effect, while efficacy tells us the maximum effect the drug can produce. These are often compared using the EC50 values. Safety index, safety of a particular compound is assessed using therapeutic index, which gives an idea of the margin between effective and toxic doses. From a medicinal chemistry perspective, one of the most important concepts is structure-activity relationship, that is SAR, which helps us to understand how the changes in the molecular structure affects its biological activity. And finally, we consider how a drug behaves in our body. That is how it is absorbed, distributed, metabolized, excreted. And now we also talk about toxic, what does the toxic effect it has on our body comes under pharmacokinetics. And in contrast to which, pharmacodynamics is the explanation of how drug behaves inside the body. What is the biological effect the drug is producing in the body? Now, I have shown in this slide, it basically summarizes the entire drug discovery journey from starting to an initial idea till the time it reaches and becomes an approved medicine. So it is a long, demanding process, typically 12 to 15 years, and it costs millions and millions to make one successful drug. Although once it is made, you can easily recover all that money also. So when we start with the target identification and screening, we followed by hit and lead generation, where medicinal chemistry plays an important role in optimizing its potency, selectivity, and safety. Only few optimized molecules, they move to clinical trials. Like in phase 1, they focus on safety. In phase 2 and phase 3, they focus on efficacy and validation before it reaches the final regulatory approval. And here I have shown a pyramid which shows that the biggest challenge is that from thousands to one which reaches the market. That is why rational drug design and precision medicines are so important. That is, they help in reducing these failures and we can deliver the right drug to the right patient. Now in this slide, it has summarized a typical in silico drug discovery workflow. How does we reach from thousands to a minimal point at laboratory level? we start with the pharmacophore model, which defines essential structural electronic features required for biological activity, such as hydrogen bonding, donors, acceptor, hydrophobic regions, aromatic rings, charge centers. These models are then used to screen large chemical database. And using this pharmacophore, we perform this database screening of virtual libraries, in which filters like Lipinski's rule of five, which are applied to ensure drug likeliness. And while this pains filters are used to eliminate assay interfering compounds, and this step dramatically reduces the number of candidates while retaining their chemical diversity. The shortlisted compounds then undergo molecular docking to predict their binding modes and their interactions with the target's active site. Then molecular dynamic simulations are used to assess the stability of these complexes over time. And in parallel, we also have ADME and toxicity predictions to eliminate candidates with unfavorable pharmacokinetic or safety profiles. And finally, the most promising molecules, they progress to experimental validation, where we synthesize the molecule, we confirm their structural confirmation is done by spectroscopies, then biochemical assays are performed in vitro, cell-based studies, and eventually in vivo evaluation, which are then further confirmed by computational predictions. What we had predicted before, they are all again validated after the experimentation. So overall, this workflow, this highlights how computational methods acts as a decision-making funnel, and they reduce cost and time by ensuring that only most promising drug-like candidates reach the experimental testing. So this is a brief about how we start in medicinal chemistry till we reach a experimental point. So now let us talk about where do we get the hits and leads from? The nature. The nature is our ultimate chemist. We all know that most of the drugs, as the data predicts, 50% of all the drugs and more than 60% of the anti-cancer drugs which are currently in use originally came from natural sources like plants or marine life. Nature gives us the lead, but we, the chemists, we turn it into a drug. Because these natural products might have some limitations, and exploring too much of nature is also destroying, it directly destroys the nature. So we as the chemists synthesize new routes in the lab, and we try to improve their properties, which allows us to manufacture them in large quantities artificially, and we get the medicine without having the harming or without depleting our natural resources. And here I have discussed two examples I've shown. One is of Taxol, which we know as an anti-cancer drug, which was used first in 1992. It was isolated from the bark of Pacific yew tree. Its first total synthesis was reported in 1994, which had multiple steps, and we are still working on to reduce the number of steps and produce more potent drugs for cancer, for which new therapies are coming every year. Another drug is discordamolide, which was isolated from Caribbean marine sponge, and it was also a potent anti-cancer agent. Now, as we have talked about the role of medicinal chemistry, and nature has given us the potent lead, like we have talked about Taxol. Here, we have a critical question that comes, that which part of these complexes or these structures which we get from nature are essential for activity, and which part of these structures can be modified. This question is addressed through the concept of structure-activity relationship studies. That is the SAR of the molecule. This relationship relates the structure of the drug with its pharmacological activity. So here, the SAR of this molecule, it shows that this taxial core is essential for binding to microtubules. And any major change to this ring system leads to loss of activity. This C13 side chain is absolutely critical for anti-cancer activity. Removal of this or major modification in this chain can drastically reduce the potency. So, and we have specific functional groups also, hydroxyl and amide here. They play a key role in stabilizing microtubules. And at the same time, some positions are there like C7. These can be modified easily as they don't play a significant role in the activity. Okay, so by integrating the SAR with the rational design strategies, we move closer to the precision cancer therapy, where drugs are tailored to specific molecular targets. So, as we have talked about, we have seen that nature provides us the complex and potent lead molecules, and SAR helps us to identify their structural features which are important for activity. We now discuss the different approaches in drug design. First and historical, traditional drug design was through analog based drug designing. That is, designing of new drugs by structurally modifying the existing lead molecules or lead approved drugs. That is, we know a molecule, we know the structure of the molecule, we can just change the substitution, we can change the chain or a ring. A slight change in the structure can bring a desired, give us the desired result, optimized result. Okay, in this strategy, what we do is we begin with a known biologically active compound. It can be a natural product, it can be an existing drug, and we structurally modify that, which can be, there can be two types of modifications, structural based modification, which we call structural analogs, and pharmacological based modification, which what we call pharmacological analogs. So I've taken example of morphine, in which, as you can see here, methyl group is bind to nitrogen, and it acts as opioid agonist. Whereas when this methyl group is changed with this alkene, here it acts as morphine antagonist. So structurally they are similar, except this functional group, they have a lot of structural similarity, but the pharmacological effect is different. And the pharmacological analogs are, as we can see, these three drugs, they are structurally absolutely different, but they have similar pharmacological property. Like in nifedipine, we have pyrimidine type of ring. Diltiazem, there we have 7-membered nitrogen sulfur containing ring. And here in verapamil, we have a tertiary amine linking to benzene rings. But although they don't have any structural similarity, but their pharmacological activity is similar. So now comes the idea to how do we optimize these structures, what changes, how these changes can be brought about. So to optimize these analogs in drug design, we can do that by bringing variations in the chain or in the substitution in aromatic ring. Like for example, I have taken adrenaline, in which again amine group is attached to methyl, and in isoprenaline there is isopropyl group attached, and in salbutamol we have tertiary butyl groups attached. So what difference this brings? These changes in the structure. Adrenaline show potency same for beta and alpha adrenoreceptors, while the other two, they show better potency towards beta receptors. And we have talked about change in aromatic substitution. Like we can change the substitution from ortho to meta to para position. It reduces or increases the resonating or inductive effect. And here it shows how it improves the binding affinity through it increases the activity through hydrogen bonding. Just changing the position of OH from para to meta position, it has increased the activity. Another way we can optimize the analog is by extension or contraction in the structure. Extension means increasing the chain length. That is, if we have a molecule, say containing ethyl linker, we can extend the chain to propyl, butyl, and hexyl. We can lengthen the chain, or we can also contract the chain in the similar way. Or we can also do it by ring expansion or contraction. Like we have a benzene ring in a system, we can change it by fused rings like indole, quinolines, or we can expand the ring from 6-membered to 7-membered ring, or contract the ring to 5-member. This also helps in better binding, having an extra group. So also I have shown some classical examples, like cisplatin had severe kidney toxicity, and introducing in cisplatin, replacing these chloros with a dioxospiro ring in carboplatin, it highly reduced the severe kidney toxicity. And another drug, that is nilotinib, it acts as super analog to treat leukemia to the patients which have developed resistance to imatinib. So this shows how structural changes helps us improve the properties. Another optimization in the analog can be brought through conformational rigidity. That is by giving the molecule, you're locking the flexibility of the molecule. Here, if we look at the conformational rigidity of this molecule, we have, this is a single bond which is rotatable. Here we have induced rigidity by introducing a double bond. Rigidity can also be introduced using a ring system, or bulky steric constraints can be used. They make the molecule metabolically stable. And I've taken the example of compresstatin, which is a class of natural product. Here it has two forms, E and Z isomers. And as we can see, the Z isomer is active, E isomer is less active. Why? See, since this Z bond, we call it cis bond, it binds to the consequent site of tubulin. It inhibits polymerization of the microtubules. It makes it rigid. This is a cis-stilbene type of structure. This is the key to activity. Whereas in the E form, E isomer, the trans isomer, this does not fit or locks with the receptor. Hence, it is less reactive. Another approach in drug design is the rational drug design. That is, finding new medications based on the knowledge of biological targets. In rational drug design, this is a systematic approach for creating new drugs based on detailed knowledge of the structure of biological target rather than hit and trial. It uses the structure of and function of the target, such as enzyme, protein, and how does the molecule bind to it effectively. The key steps which include are target identification, that is using choose which biological molecule is relevant to which disease. Then lead discovery, that is finding the molecule that can best interact with that target, and then optimizing them. That is what chemical optimization we can bring and that structure to improve its potency, selectivity, or its pharmacokinetic features. And to carry out all that, we can use tools like crystallography, X-ray crystallography, computational modeling, and SAR studies. Two major components of rational drug designing are structure-based drug designing and ligand-based drug designing. So let us now discuss one by one what do you mean by structure-based drug designing and ligand-based drug designing. Is basically it depends on the three-dimensional structure of the biological target to which we want to design our effective drug. So for that, what do we know? We want to know the 3D structure of the protein target, which we can find out using X-ray NMR, or cryo-electron microscopy. And then the potential drug which we are designing, or we already know, we have to optimize it to fit precisely into the active site. That is like a key into a lock. And so for that, what this increases the binding specificity, and it also reduces the off-target effects. So here I have given a representation of how does structure-based drug designing work. That is starting from target identification and preparation. That is, target molecule is expressed, it is purified, and it is characterized using molecular biology techniques. Then structural determination of the target is done. That is 3D using X-ray crystallography, NMR, etc. After which we perform molecular modeling. like computational tools are used for which ligand design and optimization is done through virtual screening, through docking. Then, after multiple ligand keys are tested, which best fits into the lock, we come to the synthesis part, in which the lead compounds which are selected, they are synthesized in the laboratory. Their structures are validated through spectroscopic techniques, and further the drug target complexations are again analyzed by crystallography and NMR to confirm their binding mode. Next, they are evaluated for toxicology. Okay, and then the formulation, that is the optimized drug candidate, it is prepared into a suitable dose form, and finally it reaches the clinical trials. Another type of rational drug designing is ligand-based drug designing. It is just the opposite of the structure-based. You can say that, we can say that, because here the structure of the target is not known. Where the molecule is going to bind, we don't know that structure. And so in this approach, the information is only about the active molecule, the ligand which is available. So we analyze the active molecule, we identify the key features of that molecule which can be responsible for biological activity, and then we design the new molecule of similar activity. And the techniques which can be used are QSAR, that is quantitative structure activity relationship. It is a mathematical model which links chemical features to anti-cancer activity, or pharmacophore modeling, or virtual screening. So the advantages of ligand-based drug designing is that when we are not aware of the target structure, it can rapidly generate new leads based on the existing knowledge of existing active molecules. Now that following that, we have discussed SAR analysis and we have discussed analog and rational based designing. The next stage is how we move from hit to lead in drug discovery. So as I have shown here, multiple hits are generated from screening. They are refined into fewer and fewer through high quality lead compounds, which then enter into lead optimization and which further move towards clinical to be clinical candidates. The goal, what is the goal of this hit to lead optimization, is that it has multiple parameters, not just for improving the potency, but also keeping in mind various biological properties like selectivity, minimizing off-target effects, and also having improving ADMET properties. This stage is fundamentally, basically, SAR-driven, structure-driven, and we should know how structure is contributing to the activity. Then ideal lead is, therefore, it combines high target affinity, high moderate molecular weight, and lipophilicity. And whatever lead we reaches to, it should have a practical synthetic feasibility. The route to synthesize that molecule should be feasible. To explain this hit to lead discovery, I have taken one example. This is a real example. This experiment was performed in California Institute of Technology and Kansas University. This has shown the development of CB5083, which came to clinical trials. What they did was they had a large library of around 2 lakh molecules, which was screened through HTS ATPase assay, that is high throughput screening assay, leading to 925 primary hits which were identified. And these hits were again retested for the same assay, and they narrowed it down to 333 active molecules. And further, they expanded the orthogonal assays, manual validations, and to remove false positives, all that, and it yielded these 10 high quality hit compounds, and their IC50 values are shown. So these are chemical diverse hits which represented the starting point for the SAR analysis, where the medicinal chemists identify active scaffolds, understand their key functional groups, and decide which hits are suitable for hit to lead optimization. This shows the screening funnel, how large chemical space turns into a small number of validated hits. Now, out of those 10 hits, two were selected based on the micromolar ATPase inhibition and tractable heteroaromatic scaffold. These hits then served as a core structure for SAR exploration. Here, this shows how the key structural region targeted are which were modified were this aromatic core, these substituents tertiary amine R1 and R2, and this substitution R3 region, allowing them to tune the potency, binding interactions, whatever can be improved through. So they reached to a compound ML240, 240, and this is the synthetic scheme, which shows how an efficient rule of rapidly synthesizing these molecules, in which the dichloropyrimidine ring is there. This reaction is carried out, and overall, this demonstrate that rational SAR and synthetic accessibility transforms hits into a viable lead. And now, by using this ML240, they optimized it and they made it into CB5083, which has improved properties and had gone into clinical trials. What all they had using this ML240, they had the chemical starting point, and by structure guided lead optimization, they improve the binding interaction with the ATPase, reduce its lipophilicity, they enhance the oral bioavailability, and we can see that IC50 value of this was 815 nanometer, which reduced to 11 nanometer, maintaining the molecular weight and C-log P values. It was the first in class orally available P97 inhibitor. It advanced to human clinical trials. So as now we have talked about medicinal chemistry, drug design. So what is the precision and personalized drug? As we know that interpatient variability often causes the same drug to have different efficacy or toxicity profiles. This precision and personalized medicine, this aims to overcome this by integrating patient-specific molecular information into drug development. So here, this first panel, it shows drug discovery and multi-omics profiling. This is the process which starts with drug discovery, target discovery using multi-omics profiling to analyze genomics and proteomics data assisted by AI algorithms to identify driver mutation, gene fusion. These molecular signatures, they allow to predict drug and target interactions, its druggability and potential resistance mechanism. We define this as the molecular vulnerability of the disease at individual level. Then this is the part which the medicinal chemists play, that is the rational drug designing and functional validation. Once the target is defined, rational drug design comes into play. The medicinal chemistry and computational modeling, this helps us design high affinity ligand that interacts with the target, same as lock and key optimization. Diagnostics, biomarker profiling, and pharmacogenomic data to select most effective therapy. That is the ultimate goal of this precision therapy, is right drug to the right patient at the right time. This workflow, this demonstrates how a medicinal specific drug discovery. So as now we have talked a lot about medicinal chemistry, drug design, structure activity relationships. So we have targets for cancer therapies that can be enzyme-based target therapy, and it can be immunotherapies, it can be combined therapies. So what are target therapies is the type of cancer treatment that So I will be discussing a few of these. Like I have talked about tyrosine kinase inhibitors. Mostly, I will be explaining how the structure part plays the role. We have taken example of osimertinib and ivazanib. Osimertinib is a third generation drug, oral EGFR TKI specific. It binds irreversibly to mutant receptor via covalent interactions, offering enhanced potency and reduce off-target selectivity. If we talk about its structure, activity relationship, it has acrylamide warhead. This acrylamide amine, aniline ring. It is meta-substituted aniline ring. So it reduces the metabolic degradation, enhancing the stability of the drug. And this pyrrolopyridine scaffold, this has, since this is a third generation drug, earlier drugs have indole. So it had shown better, superior activity as when we compared it to indole. Similarly, we have tivozanib here also. It has quinoline core and it has urea type linkages, which enables dual hydrogen bonding with VGFR2, providing strong kinase binding. And incorporation of this chlorophenyl ring, this facilitates hydrophobic interactions, improving the binding affinity. Another targeted therapy is the kinase inhibitors. Main structural features include hydrogen bond donors, acceptors for hinge binding regions. Here also, if I talk about their structures, I have taken two drugs, palbociclib and abemaciclib. Palbociclib, it is characterized by the presence of this piperazine ring, which significantly improves the aqueous solubility and it helps in salt formation for formulation purposes. It has a tertiary amine in this piperazine ring. This enhances oral bioavailability. And this pyridine ring, which is attached to this piperazine, this is crucial for binding CDK selectivity. And this cyclopentane ring, it contributes to hydrophobic site filling, enhancing CDK6 inhibition. So now about another therapy which I have discussed is combination chemotherapy, which is the use of multiple agents with different mechanisms. The combination of sotorasib with vasitibinix is used. That is, it is the combination which was approved in early 2025 for the treatment of adult patients with metastatic colorectal cancer, but who have already prior received chemotherapy. It is this sotorasib. This is a covalent inhibitor targeting the rat sarcoma gene mutation, where the amino acid glycine is replaced by cysteine mutation. While this the drug with it is combined is vasitibix. It is an anti-EGFR monoclonal antibody. This dual targeted approach, this addresses the activation of EGFR signaling, and it often limits the efficacy of class inhibitors. If I talk about the structure of these two drugs, here the sulfonamide, this group sulfonamide, it enhances the binding by hydrogen bonding, and the lipophilic tail contributes to this is the lipophilic tail. This contributes to improved cell permeability and oral absorption. In encorafenib, the pyrazolopyrimidine, this rings this scaffold with sulfonamide. This helps in targeting the ATP binding. Then let us talk about some FDA-approved drugs into clinical trials. They are targeted small molecules. They are designed for specific oncogenic target. Here I have discussed, given the structure of two, divarasib and glacirasib. Divarasib, they are class inhibitors. And if we talk about their structure activity, relationship of this divaricib. It has highlights the importance of this 2 amino 4 methyl and this 5 trifluoromethyl, this ring, pyridine ring, this moiety, and at C7, this enhances the binding of cross switch pocket. This properly orients with the acrylamide, this warhead. It acts as a warhead for covalently targeting the cysteine. This also maintains the binding pose across the GTPase supporting scaffold and flexibility. These findings emphasize the need to optimize both reversible binding and warhead reactivity for selective class inhibition. So there were changes. It had certain drawbacks, so the changes were brought about in the new drug, that is glaciracib. In this, this is another irreversible class inhibitor. This is in clinical development. Um, it has selectively binding to the and stabilizing GTP bond and to the inactive form of cross. It has this 1,8 naphthalidine 3-carbonitrile. This is 3-carbonitrile scaffold, which anchors to the switch pocket 2. While it is in position with this warhead again, which binds to cysteine 12. Further, this SAR refinements improve the solubility, reduce the metabolic liabilities, enhance their pharmacokinetic properties, resulting in a potent, well-balanced clinical candidate for class-mutant cancer. This glaciracib, this represents an optimized inhibitor with nano molar potency, and it increases its selectivity to 500 folds, contrasting with the earlier leads. Then let us talk about a new approach that is trending latest, the PROTACs, that is proteolysis targeting chimera technology. They induce the target protein degradation instead of inhibition. So I have taken the example of galutamide. This is a novel AR degrader which uses a PROTAC technology. This PROTAC-based AR antagonist and a degrader, this was developed to overcome the resistance in metastatic castrate-resistant prostate cancer. Here, the PROTAC, this is formed by combining 3 optimized components. The 3 components are the high affinity AR ligand, that is, and then the E3 ligase recruiter. E3 ligase recruiter, this give the efficient degradation, influencing the kinetic and off-target effects. And then this optimized linker, its length and its polarity, this influences the ternary complex formation and the cellular activity. So this is all about the type of therapies, cancer targets, which are currently in use. This shows the structures of the FDA-approved drugs. I've taken examples of tyrosine kinase inhibitors here. As you can see through the structures previously discussed and this slide, that there are heterocyclic rings which play the key role. Especially we have seen nitrogen-containing heterocycles, pyridine, piperazine, morpholine. So it is observed that heterocyclic rings a crucial role because they also help in hydrogen bonding easily. They have a lone pair of electrons. Keeping all these points in mind, we medicinal chemists design our molecules. So now here I'm going to present one of my molecule which was designed in my laboratory, that is hydroxamic acid-base carbonic anhydrase inhibitor. This was designed by combining the features of known clinical candidate, this SLC 111 and some maplin C. So from here we have chosen this part, the zinc binding group, which were, and here the oxime hydroxamic acid group was introduced. This group acts as a claw that grabs the zinc ion in the enzyme's active site, which helps in effectively stopping its activity. We have this linker and this tail. This helps the drug to fit into the enzyme and stabilizing the binding and improving its potency. So the synthetic route, keeping all these points and expecting it to show good results, the synthetic route for this was designed, and these compounds were synthesized in my laboratory. There were four key steps. Uh, First, we had taken isonicotinaldehyde, that is pyridine carboxylaldehyde, which reacts with hydroxylamine in presence of ethanol for 10 to 15 hours to produce this enzyme, which was further reacted with N-chlorosuccinamide NDMF to give chlorosubstituted oxime, which further reacted with these piperazine sulfonamides to give the target molecule. Here we had sulfonamides, here we have oxime and this pyridine linkage and this linker. 10 such derivatives were synthesized in our laboratory. Not alone this, similar type of compounds containing indole and sulfonamide linkage, which are linked via chalcones, such compounds were also synthesized. Another series was synthesized containing hydrozoles, in which indole and sulfonamide were linked through NNH bond. And morpholine was used instead of piperazine here. And these were the one which I have shown the synthesis of. So these compounds had shown they were potent towards cancers associated carbonic anhydrase enzyme. They showed good toxicity, cytotoxicity profiles, binding affinities. The results were quite good. The cancer studies were performed on HEK cell line and MCF7 cell line, breast cancer cell line. And we have published 3 papers in ACS journals from these 3 series. So to conclude, I can say that to sum up all these studies, can say that the new study that highlights how the structure-based medication design can drastically speed up the process of finding and developing new drugs. And using these comprehensive structural knowledge and biological target knowledge, structure-based drug design, it provides a logic and an effective method for creating new medicine with improved potency, selectivity, and safety profiles. So I would like to conclude as as medicinal chemistry provides a molecular framework for precision and personalized medicine by enabling rational and target-specific drug design. Advances in structure-based and ligand-based drug design allows the development of therapies tailored to specific molecular targets, pathways, and disease subtypes. And precision method shifts from one drug fits to all to right drug to right target to right patient, improving the efficacy while minimizing the toxicity. And overall, the medicinal chemistry plays a central role in translating the biological insights into safe, effective, and personalized therapeutic agents. Last but not the least, I would say the precision medicines begin at the molecular level, but rational drug design is its driving force. Thank you all for giving your time and listening to me. I think I finished quite early.
It's absolutely fine. Thank you so much. We can take some questions here, right, Swati? We have got some time.
I think I either they are already well aware with the topic, as they have been involved with this from past 4 days, or everything was beyond the
It
scope.
was very
So
clear.
I had actually kept it to the basics only.
Yeah, but I have seen that they warm up a little bit later, so don't worry. You might see the questions later on, and you can answer them as we go. So we will start at 12 with the my lecture. I'll put up my current slide, and Please feel free to ask any question that is related to the subject, or this is also an opportunity for asking a career level question, because we have got some undergrads here as well as graduate students, so you can ask. And this was a very befitting lecture, Dr. Afreen, because we had a recently concluded Gyan course, which was on drug discovery and delivery. So your lecture connected the two Gyan courses that we conducted here. Um, Thankful to you for that. I think there's a question here. Swati, can you take it up?
See, what we can say in personalized cancer therapy. See, as I have explained already, drug design has played an important role, and a molecular this heterogeneity. When we get into the depth and we relate the structures to whatever type of cancer it is, we come to know the optimization the structure can bring. We can optimize the structure to personalize it to a particular therapy, depending upon the different targets and the already present drugs. See, I am not into much into the biology of all these. I am a chemist, so I think I can just say that combining the knowledge of both the drug and the target can help you in this to get a better personalized drug for cancer.
I can take up that question, Nilofer, because the molecular heterogeneity affects the finding of the target. That makes the life of the hunters for these cancer targets difficult. As I told you in my lectures, these are moving targets, plus they are different from one person to another person. But once the target is identified, then the drug design is not designing its drug from scratch for that particular person. Rather, we try to see what has already been designed and what has already been available as FDA-approved drugs or libraries that we can bring into for helping this particular patient. Tumor heterogeneity or molecular heterogeneity is a problem for finding cancer drug targets, but once target is found, there's no problem with the drug. Unless there is something very, very rare, that is that there is no drug or molecule available to target that. Does that answer your question, Nilofer? Right. So I think we will just take it.
I think it's already 12 for 3-4 minutes. You can take a tea break or something if you want. Thank you, all of you. I'll just take your leave now.
What's left? We have been sticking to the schedule.
Thank you so much. Thank you, all of you. Thank you for, I would like to thank Professor Azhar Aziz for giving me this opportunity to speak today in front of you, all of you. Thank you, sir. And thank you for answering that question also on my behalf. Thank you.
There's another question by Fatima. Can you take that?
Yeah. How do you select the lead compound and what factors are? Okay. See, I have already discussed, we decide the lead compound by screening through computational studies. That was there in my slide in the very beginning. I said, we, from the library of compounds or from the already drugs which are the nature has given us, or we have already we have so many drugs in the market, medicines in the market, they act as a lead for the future designing and optimization of the compound. Like I have taken one example of Taxol, that it was extracted from plant and later on its total synthesis was designed, and now also whatever drugs for a particular disease or an infection is present in the market, we can take it as a lead compound. Or I would give you an example like for malaria, uh, the plant quinine was used from the bark of that quinine was extracted from some plant. Uh, so later on, when we develop the drug for malaria, whether you talk of chloroquinoline, amodiaquine, all those have structural features similar to quinine, which was extracted from a plant. So nature, first of all, nature acts as a base to start from. That is from where we start. And the drugs which are already in market and the drugs which have already been synthesized, which are FDA approved, they set the base to new lead optimization. I hope I have answered. Thank you. Anyone, any more questions, please?